Hi, I'm Ushma Neal from the Journal of Clinical Investigation. I'm happy to have Dr. Eric Olson of the University of Texas Southwestern Medical Center in the studio today for the next in our series of Conversations with Giants in Medicine. Though when he agreed to sit for this interview, he asked me to change the title to Conversations with Mere Mortals in Medicine. Olson is best known for his pioneering research on the molecular and genetic mechanisms of cardiac and other muscle development and regeneration. He's also very well known as an accomplished musician, and I hope over the course of this conversation to probe these multiple motivations. So thank you so much for joining me today.
Thanks, I'm happy to be here.
Great. Can you start by telling me a little bit about your parents and where you were raised and what you were like as a kid?
Yeah, sure. I was born in Rochester, New York. My father grew up on a Norwegian farm in South Dakota without running water and without electricity. And he escaped as soon as he could to go to college, and he became a chemist. So my father worked at Kodak in Rochester, New York, and there he met my mother. My mother was a musician. She and her mother before her, they taught piano and music theory at the Eastman School of Music. So I was raised in Rochester during my early life, and then my father was a bit of an adventurous spirit. So we moved to North Carolina when I was in grade school. I lived next to the campus, Wake Forest University. And then as I got to high school, we really started a nomadic existence. I went to the 10th grade in Richmond, Virginia. I went to the 11th grade in Fort Worth, Texas. I went to the 12th grade in a small town in upstate New York called Norwich, New York. So by the end of my high school experience, I was ready to find some familiarity again. So I applied to Wake Forest and went back to where I was most familiar. Stayed there. I majored in chemistry, biology, and I even stayed on there and got a PhD.
So what were you like as a kid? Were you mischievous? Were you a good student? Were you boisterous?
I was probably an average student. I was always interested in science. I mean, even from grade school, I remember when the class would go to the library and the teacher would say you can all spread out and go find the types of books you're interested in. I would always gravitate to the books about science. So, I always knew I would go into some sort of a career in science. But coming from a musical family, music was always a part of my life growing up. I started out playing piano as a young child, and then I played trumpet all through high school in the symphony and jazz orchestra, and then later picked up other instruments. Yeah.
So let's probe a little bit of the interest in science. Did you have any particular mentors, or was it simply the fact that your father was a chemist that kindled an interest in science?
I think my father certainly kindled that interest. I was drawn to science, but more to the aspect of science about learning things new. I was always interested in the edge of knowledge. I was never so interested in memorizing facts. And it's interesting, when I got to college, I knew I was going to go into a scientific career, and I double majored in chemistry and biology. But because my high school years had been kind of disruptive, moving around a lot, I hadn't had a real continuous education through biology and chemistry. So I struggled a bit in my first year of college in the science courses, and I remember my first year advisor called me in, and she sat me down and she said, Eric, we need to tell you, you don't have a future in science, and you need to start to think about an alternative major.
Hot dang, and so look at you now.
So, well, that was very motivating, and it really encouraged me to redouble my efforts.
Okay, so you decided that you were still interested in biology and you were going to keep going regardless of what this mentor said to you. Then how did you decide that you were going to do a PhD?
Well, I finished college majoring in biology and chemistry, as I said, and I had never, for whatever reason, been interested in applying to medical school. As I said, I was more interested in discovery-based science and really the freedom to explore things. And so I decided to stay on at Wake Forest because I was comfortable there and get a PhD. So I did that, and then as for my PhD, I worked on skeletal muscle membranes and analyzed phospholipid changes that took place in the membranes of muscle during diabetes. It was a very biochemically based project. And so then as a postdoc, I thought, okay, I've got to move on now. There's not much left for me to do here. So I applied to WashU, and I worked in Dr. Louis Glazer's laboratory, and that was a real eye-opening experience for me. WashU was a wonderful scientific environment there, and I felt like I'd kind of gone from the B team into the big leagues to be there. I had a lot of freedom, and as a postdoc, I thought about what I wanted to work on, and I developed a project that kind of connected my prior work with my interests, and that was I studied how lipids were attached to membrane proteins, and in particular I studied one membrane protein, the acetylcholine receptor, which is a muscle-specific membrane protein, and that really got me interested in the processes of muscle differentiation.
Okay, so at the beginning it's more biochemistry, but it happened to be skeletal muscle. Do you think that that was ever something that you said, yes, muscle biology, this is where it's at?
Well, so at every step in my career I really thought strategically about what I wanted to do. So when I finished my postdoc, I applied for a couple of positions. I didn't really apply for very many, maybe a handful. Louis Glazer, my mentor, told me that a good colleague of his, William Lenars, was starting a department of biochemistry at MD Anderson Cancer Center, and he thought that would be a good environment. So I applied there and was fortunate they hired me. And so at that point I knew I've really got to think about what it is that I'm going to do in the future, and how can I carve out my niche, and how can I pick a topic that will sustain me for decades. So I knew about muscle differentiation, and that was a time when gene regulation was just beginning to be studied. Transcription factors hadn't even been cloned then in the early 1980s. And since I knew about muscle, I decided I wanted to study how muscle cells differentiate. And part of that was pragmatic, because you could get a cell line of muscle cells inexpensively and study them. And when they differentiated, hundreds of genes would come on. And so it just seemed like a perfect model to try to probe those, the questions of how batteries of genes get turned on in differentiation. There was only one problem. I had no experience in molecular biology, and none of the previous labs I'd worked in were molecular biology labs, and so I had to pretty much teach myself from square one how to do that.
And so when you started your first lab, was that a successful experience? How was it getting grants and falling into a completely new field?
I was naive. Maybe fortunately I was naive. I didn't know how difficult it would be at the beginning. I didn't come, as I said, from a background in the muscle field, and I had no experience in gene regulation. But I decided, okay, I'm going to start simple. I'm going to try to just figure out what DNA sequences turn on one muscle gene, and let's see if I can figure that out. I bought the Maniatis cloning manual and started learning how to do this, and I selected a gene called muscle creatine kinase, or MCK. So I wrote my first NIH grant on this. It got completely demolished. That was a little demoralizing. So then I revised it. I wrote it, sent back the revision. It got demolished again. So now I'm about 3 years in. My startup funds are running out. And so I tried one more time, and I finally got my R01. That was sort of the last gasp. And then really the momentum started picking up. I was getting some good students. I had some amazing postdocs too at that period, which came from all over the country. These were young people, and it was gratifying to me that they would even bet their careers on me. Many of them are famous people now in the field, and so we really got an energy vibe going. And then just as things were really coming together, my department chair, Bill Lenars, accepted a position at Stone Kettering, at Stony Brook. And so he moved to Stony Brook, and he asked if I would come with him to Stony Brook, and they had a Howard Hughes position there actually that I could have had if I had moved there. And I thought about it, but I said, you know, I just spent all these years getting my R01 grant, built this group of people, things are coming together, I feel really good about this, I'm just going to take my chances and stay here and keep going. And so that's what I did, and ironically, a year later, I became the chairman of the department.
Right, so I was going to ask you about that. You're 35 years old, you're finding your way through a new field, and you find yourself also as department chair. So how did you deal with the administrative burden? And I mean, fast forward to right now in 2016, would that ever happen again, that we would give the chairmanship of a department, especially of molecular biology, to a 35-year-old? And should we?
Well, it worked out pretty good in that case. Those were different days. It's remarkable now to realize I was 28 years old when I started my own lab, so
Wow.
I was pretty young. And so by 35, I was a department chair, and that wasn't something I set out to do.
Mm-hmm.
And when they first approached me with that opportunity, I thought, this is something I've never even thought about doing. But then I really got excited about it because I felt like I could create an environment the way that I would like to see it created, and I could recruit people and interact with people in an intimate department setting. And so it really turned out to be a great opportunity for me.
Have you enjoyed being a chair all this time?
I've been a chair now for about 26 years. I can't say I've enjoyed it all the time.
Okay, so you moved from MD Anderson to University of Texas Southwestern in Dallas, and you've done something different from when you were raised. You've stayed in the same place for years and years and years. Why stay? Because I know you've been offered many opportunities to leave.
Yeah. Well, maybe that could still be a hangover from my younger days when I was tired of being uprooted and moving around a lot. But really, I love it in Texas. I like to come out to the coast. There's a lot of energy in New York and in Boston and California. And obviously, those are the epicenters of science. But I find it so refreshing to be in Dallas, sort of away from the crowds. I feel like I can think out there, and I love the big sky and sort of this pioneer spirit that we have. And I think, in Dallas for sure we have that spirit. I think a lot of people in Dallas who are the true giants of medicine, as well as myself. We've planted our flag there, and this is the place that we want to make great. And so I'm really proud of being a part of that.
Nice. So over the early course of your career, you made your name identifying a lot of the different transcription factors that cause the differentiation of muscle. But one of the major discoveries you made was towards cardiac muscle. And that's what you've been known for the bulk of at least our own interaction. I've always known you as the big heart muscle guy. So at what point did you pivot towards the heart, and why have you not necessarily pivoted away from that?
Well, it goes back to the strategy of the onset of my career. When I first started working on skeletal muscle, I thought to myself, maybe if I could discover a couple of interesting genes in skeletal muscle and learn something about muscle, that maybe then I could leverage that knowledge to move towards the heart, which is the most interesting of all, but it was completely intractable in the early years. And so once we got traction in skeletal muscle, and my students discovered many of the key genes that orchestrate the formation of muscle, and once we had laid out that framework, then we moved to the heart, and we discovered some of the key early genes in heart development.
MEF.
The hand genes,
Yeah.
well, MEF2 we discovered, of course, and the hand genes, which make the left and right ventricle. One of my close friends and colleagues, Deepak Srivastava, played a big role in that early work. And then we went on and discovered myocardin, a master gene for activation of sarcomere genes. And so that's how it kind of evolved from muscle towards the heart. I still am interested in skeletal muscle, and we still do a lot of work on that. I mean, I think the whole field of muscle biology has been really fertile ground for discovery, and that's what I like best. I like to really operate at the outer edge of knowledge. I'm more interested in discovering my own things, not working on other people's things. I want other people to work on my things. So, and so the muscle field's been a great area for discovery, but also it has so many connections to disease. So along the way, we've been fortunate that many of the genes and the regulatory mechanisms that we discovered had very obvious connections to diseases of the heart and of muscle.
You started off making a lot of basic biological findings, but you've pivoted towards some regenerative medicine, including a CRISPR-Cas9 model for repairing muscular dystrophy. And along the way, you've become a successful entrepreneur and started 3 different biotech companies. So where did the entrepreneurial streak come from?
Well, I think it just came from an adventurousness, I think, and I think I got that from my father. He started many different companies when I was growing up, and I watched that, and he mentored me a lot in how to start companies. But I just always wanted to see our work push as far as it could, and to go beyond basic science. And it became clear as we were working on the early heart gene regulatory programs, particularly when we were studying histone deacetylases and how they're regulated by various kinases, that there was therapeutic potential there. And so I was involved in starting a company called Myogen Therapeutics, co-founded it with 2 close friends of mine, Mike Bristow and Leslie Leinwand, in Boulder, Colorado. And the work from my lab really became the foundation of that company, and it also served as an outlet for people from my lab who were interested in biotech, and they would go, several went to the company and continued the work from my laboratory there. And I just found that to be so interesting at every level, including coming to Wall Street trying to raise money when everyone said it wouldn't work, to just the people I was able to meet, and just to see how you can transition from academia to industry. The process is completely different. I mean, in academia, everything is interesting. You can just go in any direction that you're interested in, and any discovery at the edge of knowledge is important. But in a company, of course, it's got to be really focused and practical. So that's what got me very excited about that. And Myogen ultimately was acquired along the way, and so then I was able to start another company, Miragen, sister of Myogen. It's a microRNA-based company, very exciting. They've got some very exciting things going on there now, therapeutically. And I'm starting a new company right now to address the issue that you just raised, and that is, can we use gene editing to permanently correct the mutations responsible for muscular dystrophy? I am so excited about this project, I got to tell you. So we've been able to deliver CRISPR-Cas to muscle, and with a very simple gene editing step to bypass the mutations responsible for Duchenne muscular dystrophy. There are about 3,000 mutations that have been identified worldwide now that cause this disorder. And we think this strategy can correct maybe between 60 and 80% of those mutations.
Wow.
So it works beautifully in a mouse, of course, you know, if there's a big step from a mouse on, but we're trying to push that forward, and I'm really committed to seeing if I can do something, and I have my eyes wide open. I know the challenges, but I think this is a really exciting time for bringing the technology of gene editing, which was obviously developed by others, to an area that I have a lot of expertise in, and that's in muscle, genetic diseases of muscle, and to see if we can do something.
So You mentioned something earlier, and it rings true for in everything that I read about every different major award you were collecting along the way. You were always very quick to acknowledge all of the colleagues and trainees from your lab that helped you to get to the findings that you have. And in fact, I heard that you were recently awarded the American Heart Association Braunwald Lifetime Achievement in Mentoring Award. And I took the time to call up a couple of your former trainees to ask them about what it was like in the Olson lab. And two of them said, listen, this was not a place to have your hand held. This was not a place to faff about with a bunch of different ideas, that you were very driven, but that you helped them suss out exactly what was a good idea and how to drive towards that goal. They also said that it was a place where you weeded out a lot of people who shouldn't be academic scientists. Like if they could hang in your lab, they were probably going to be successful. So what is your approach to mentoring, if I didn't just take the words from your mouth?
Well, I mean, you said it. I've been really fortunate. I had a lot of talented individuals from very early in my career, when I was a complete unknown, to bet their career on me, and I take that really seriously. I think I've been lucky that there's a filter for me in Texas. Anyone who comes to Texas to work with me, I know they really want to do science. They don't want to go surfing on the weekend. I'm a very engaged, hands-on mentor. I mean, I spend at least half my day every day in the laboratory. I try to talk to every person every single day. I work on the weekends. That's the time when I can really talk to people. So I'm really engaged, not micromanaging, but just there to express my enthusiasm, which is authentic, and to find out what they're doing. So I'm really into it. Mentoring is, I see mentoring as really a way to enable the trainee to find their best path to fulfillment. When people are trainees, they're in a formative period of their career development, and oftentimes, most of the time, they don't know exactly where their strengths and weaknesses are. They don't know exactly how to move forward, but they want to be successful. And so I think one of the challenges of the mentor is to figure out how can this person achieve their greatest potential, and that involves figuring out what is the risk tolerance of an individual. Some people can handle risk. They like to discover things and work without a safety net. Other people would rather work, dot the I's and cross the T's on a project. There's no one way that's better than another way, but you really have to match the trainee to the project to enable them to be successful. Another thing that I have tried to do is I give everything away. So when a postdoc in particular, when they come to my lab, I encourage them to find their path, and I work with them to do that. And if they make a great discovery, it's all theirs. They can take it with them. I'm not going to compete with them, not that I could anyway, but I try to let them have as much room as they can to develop, and I think has ensured their success. It also had a reward for me that I didn't anticipate, and that is when you give things away, it forces you to keep growing and learning, and so you don't become stale. So I'm always having to keep trying to find the next big thing in my field and try to push the field forward, because otherwise I'm trying to compete with a former postdoc, and I can't do that.
So do you think that there's enough low-hanging fruit out there for you to be able to keep doing this in terms of muscle and cardiac development and regenerative potential?
Well, I have to say everything we're doing is not low-hanging fruit. There's some pretty high-hanging fruit that we're going after. I mean, I think so far I've not run out of things to do. I haven't run out of ideas. I'm as excited now as I was 30 years ago about some of the things that we're working on in my lab. I hope I have the energy to keep going for as long as possible. But right now, I'm really into it, yeah.
Well, cool. So I want to pivot towards the musical side of you. We've talked about your father's influence on what you have become professionally, but clearly the maternal side and the piano that you started from the beginning has also had an impact on you. So you play guitar and harmonica and sing in a band. So what side of you, what itch does that scratch, being musically inclined as well?
Well, I love every aspect of music, and I always have, for whatever reason, there's always a song rattling around in my head. So I just love music. I'm not an especially talented musician, but I really enjoy it. So that gives me a creative outlet. And one of the things that I'd never done, though, was to play in a rock and roll band. It was always something I wanted to do. So when I was approaching my 50th birthday, which was almost 12 years ago now, I decided, okay, if I'm going to be in a rock and roll band, I better do it. So I was fortunate that there were some really good musicians in my department and around UT Southwestern, and so I got them together and we started this band called the Transactivators. It's been awesome. We play all over the place, and it's so fun. So I've found that to be a creative outlet, and actually it's funny, I'm kind of a reserved individual, but when I'm with the band and we're up there plugged in and everybody's playing together, you just forget about all that, and it feels like this is like my alter ego. There's no self-consciousness when you're playing in a band and the band is really in the groove.
Well, I think you're being shy. I've seen some of the videos. I've actually never gotten a chance to hear you play live, but I've seen the videos and I think you are quite a good musician. Now, one of the other things I remember you telling me, it's the one thing that everybody asks you about, and that is the fact that you are the Annie and Willie Nelson Endowed Chair at UT Southwestern. So how did that come about? Because it's fascinating for the rest of us.
Yeah, well, that's actually, it's humbling to realize that's probably what I'll be remembered most for. Not my discoveries, but being the Annie and Willie Nelson Chair. I mean, Willie Nelson is a national treasure. And coming from Texas, he epitomizes Texas. He's the greatest icon of Texas. And I had always adored Willie Nelson's music. Well, it turned out that he and his wife, Annie, were bringing their two sons to Southwestern for their checkup. This was so many years ago now. And Kern Wildenthal, the president, I guess he wondered what to do with them. And anyway, he knew that I was into music, so he arranged for me to have lunch with them. And we really hit it off. And so then Willie Nelson had a benefit concert and raised money to support the research and to create this Annie and Willie Nelson chair. And then along the way, I've had many interactions with him on his tour bus before concerts or at his ranch west of Austin. And
have you ever gotten to play music with him?
I have not played music with Willie. I should do that, but I have not done that yet. Maybe that's going to be my next goal in life. But yeah, being connected to one of my all-time heroes and musical icons, Willie Nelson, has really been a thrill.
Do you have a favorite color?
I think the one I'm wearing. My wife says I look best in blue, so.
Well, and so then I asked your trainees this. They both said that you liked blue, but they also said they had no idea if your hair has ever had color. Were you born with white hair? I've known you for 15 years, and I've always known you with this shock of white hair.
I had dark brown hair.
No.
Dark brown curly hair until my early 20s, and it started to go. I think this is the Norwegian side of my family. My father's parents were white, and so I have that Norwegian blood, but I guess most people don't know me like that.
I think you would be kind of unrecognizable, honestly. All right, do you
I thought about coloring it, but I thought people might notice.
So, at the end of a particularly difficult or complicated day, week, month, year, have you ever contemplated a different career other than being a scientist?
I have never contemplated a career other than being a scientist. Even when I'm really frustrated at getting a paper rejected or a grant or something's not working, I really love what I do. And I didn't even know all the dimensions of this job when I went into it, but it is the coolest career. Unfortunately, young people today don't really understand it or appreciate it, but it is so amazing. It's almost spiritual, I feel, to be able to discover things at the outer edge of knowledge that no one has ever discovered, and really to lay down fundamental facts about how life works on this planet is really just an honor to be able to do that. But then there's so many other dimensions to it. There's the ability to travel all over the world and talk about things you're interested in. There's the ability to interact with young people, and in my own lab I've had people come from all over the world. We have a really international lab, and I've gotten to meet and interact with people from foreign countries that I would never have interacted with, and travel there and be part of this enormous family of young scientists that have trained in my lab. So it's just been such a gratifying career, and as you pointed out, it's also offered me opportunities to learn about business through biotechnology and to try to do things to improve human health. So I think it's great, and you hear a lot of complaining, of course, and I worry about this with young scientists, that they hear their mentors complaining, oh, it's so hard to get grants and all this stuff. And it can be, but really the upside is just, it's amazing. So yeah, I'm very happy with this career.
Okay, then I'm going to ask a different question and force your hand. If you had to pick again and you couldn't be a scientist, what else do you think could have kept you happy and motivated?
I think if I wasn't a scientist, I would certainly go into some career that had freedom to operate and an ability to create your own thing. I would not work well in a 9-to-5 clock punching mode or a well-defined path. So I might go into writing. I like writing, or I might go into some aspect of music, not necessarily performing, but some aspect of it that has a creative bent. And I mean, science is a creative enterprise. It's a really fascinating enterprise when you think about it, because it connects extreme creativity with the absolute objectivity. And that's what science is. It's the melding of those two extremes, probably more so than any other profession.
So it always just comes back to science then.
Well, I'm a science nerd. What can I say?
Excellent. All right. Well, then it has been such a pleasure hearing more about the Eric Olson with dark hair and and the one that you developed into. So thank you so much for joining me today.
Thanks for having me. I've had a great time. Thank you.
Is everybody having a good time out there? Mom, who never was around, but she grew up tall and she grew up right with the Indiana boys on the Indiana